What do you do when something bad comes to your way? Whether you move away from it or just find a way to escape it. But imagine being in a situation where you cannot move and dangerous things are coming your way that can harm you or even worse, eat you. Plants being sessile are always in that position in which they cannot escape environmental extremes. But they still need to survive, and thus they have to find a way to be well adapted to local environmental variation. Have you wondered how? And here epigenetic comes to play a role. Epigenetics is the reason that can do modification in gene expression of organisms without altering the genetic code itself. So this is an added layer on top of genetics that can explain many things, such as plant response to different stresses. There are growing evidences indicating that epigenetic regulation can modulate plant responses, including priming, which is the ability of plants to mount improved future defense responses following herbivore or pathogen attack or exposure to particular environmental conditions. Also, we could say it is like us remembering a pain and getting stronger after we suffered from something. But still, our understanding of epigenetic contribution to plant-herbivore interaction is fragmentary. So in this interesting project that I'm working on, I'm trying to understand the role of DNA methylation, which is one of the epigenetical mechanisms in plant responses to herbivory using the diverse 3 focal wild plants of Epidiverse. The plants are an annual plant biofuel crop named Thlaspi arvense, an asexually reproducing perennial plant woodland strawberry named Fragaria vesca, and a long-lived tree named Populus nigra. Diversity in our wild plants gives us an amazing possibility to investigate how epigenetic mechanisms contribute to stress responses and long-term adaptation. In my work, natural insects and simulated herbivories are used as fixed treatments for all the 3 species, and for analyzing DNA methylation changes, we will be using a method called EPGBS, which is a reduced representation bisulfate sequencing method. Finally, as outcome, we are expecting to find out how herbivore stress induces DNA methylation changes in plants within population and across geographical region, and also to check offspring to know how stable the maternal resistance is if there is any. So I'm hoping this work can highlight the potential of epigenetic diversity in ecological settings as well as will provide functional insight in evolutionary process. And thank you for watching.